From the Llano Estacado to the Rio Grande Valley, from the bayou to the bottomlands, from the desert to the rolling plains, this is seminar for a wide range. Welcome to another episode of the Soil and Crop Sciences Hour of Power. Today's guest comes to us from the great sorghum / cowpea / small grain / cotton growing region of the rolling plains, and originating, I believe, from the great vanilla and lemur producing region of Madagascar. So our guest today is Dr. Waltram Ravalambala. He graduated from the University of Arkansas, Fayetteville with a Ph.D. in horticulture in 2020. His research background has been on germplasm evaluation, development, and validation of molecular markers, use of whole genome resequencing data, and model development and testing for genomic selection. He started working as an assistant professor working on organic and specialty crops at the Vernon Center on August 18th, 2020. Oh, well, he's been there for not so long, I guess. His research interests are understanding the genetic mechanism of abiotic and biotic stresses in plants and developing molecular markers for speeding up the development of crop cultivars. Crops of interest include legumes such as cowpea, I hope Dr. Singh is listening, guar, lentils, and other organic crops. He's also interested in integrating plant physiology, genomics, phenomics, and genomic selection, which can direct the cultivar development process. Dr. Ravelombola, please take it away.
Thank you so much, Dr. Finlayson, for this nice introduction, and thank you for all of you for providing me with this opportunity to present today. So I hope I am not too loud, neither too quiet, and the slides hopefully are appearing on your screen. So I'm making the slides a little bit bigger so everyone can see. So as Dr. Finlayson mentioned, I am a new professor on specialty and organic crop breeding at the AgriLife Research Center, and I'm based at Vernon. I just started the position 1 month and few days ago. And for this today's presentation, since I don't have data yet from my program, I will be presenting some data that I have some work that I've been working on before. And I picked the plant Calpe since Calpe at Texas A&M. There are great faculties that have been working on these crops. And I'm looking forward to collaborating to various faculties working on legumes and any organic crops of interest as well. For today's presentation, I will be talking about conventional and molecular breeding in Calpe. First of all, I would like to thank all the people that helped me throughout the process in terms of research and any other possible support that we can think of. And I am very grateful to the center director here at Vernon, Dr. Verling, and the department head of the soil and crop science at Texas A&M, Dr. Baltensberg, and my previous department head and advisors, as well as the faculty here at Vernon and at the college station. And I would like to especially thank Dr. Margaret Eisen Mason and Dr. John Ruber Clark for helping me as well during my graduate studies, and all the lab members and staff members from my previous university, and the staff members at Vernon and all different universities where I have been collaborating, and you, dear audience, for your time and your support for this today's seminar. It is really appreciated. So for today's presentations, I will be having some background on Calpe, then a little bit of terminology, since I'm assuming that not all the audience are familiar with breeding, molecular breeding, and sequencing. Then I will elaborate a little bit about conventional breeding in Calpe, some work that has been done, and then some not really new technology like genotyping by sequencing and whole genomic sequencing and their use in molecular breeding in Calpe. So Calpe, the scientific name is Vigna unguiculata. It belongs to the family Fabaceae and it's a diploid species with 11 haploid chromosomes. It is a self-pollinated crop. And in the United States, people know cowpea as black-eyed peas or savoury peas. And on the right side of the slide, we can see that cowpea is very diverse. It is a very colorful crop, and it has a lot. A lot of genetic diversity. Talking about the genetic diversity and genomic, the size of the Calpe genome is about 620 MB, and a large portion of it has been sequenced. And the sequence information is available on the website called Phytosome. And also there was a paper published by the scientists from the UC Riverside regarding the Calpe genome, which was published last year. So cowpea is a legume of economic importance, especially in developing countries. And this statistic is a little bit old, but I think it's still valid. It's about the production, the annual production of cowpea, which is about 5.4 million metric tons a year. So people use cowpea for eating. It is a very good source of protein. And I have heard that some people might be allergic to soybean protein. So if that is the case, we can suggest to those people to try cowpea protein instead. And also, cowpea has a lot of use. It is a multi-purpose crop. It can also be used as supplement for feed for livestock. And in terms of ecosystem, cowpea can also be used as a cover crop. That's the brief introduction about cowpea if you're not familiar yet with the crop. And I hope that this brief introduction can give you some basic idea about what is cowpea, how can it be used, and what the current status of the genetic and the genomic information. Let's now move to some terminology that I will be talking a lot during this presentation. Single nucleotide polymorphism. So it's a single variation between nucleotides between different individuals. On this picture, we have 6 plants and we have 3 different SNPs, SNP TG, SNP CA, and SNP AG. So the SNP is the single variation of nucleotides between plants. And if we look at plant 1 to 3, highlighted in green, and then plant 4, 5, 6, highlighted in blue, and on the right hand side of the slide, we can see that those plants have different fruit size. And let's look at the SNP TG. So the plants 1, 2, and 3 have nucleotide T, but the plant 4, 5, 6 have nucleotide G. And we can see that plant 1 to 3 have bigger fruit size, and plant 4, 5, 6 has smaller fruit size. So we can also associate this SNP TG to fruit size, and that's a very short story of what we call association mapping or genome-wide association study, or GWAS, as you can see at the bottom of the slide. And of course, there are other factors that we should consider when doing this analysis. And we have done a lot of similar analysis in Calpe that I will be elaborating more later. One of the new tools that is widely used right now, and especially in crops such as wheat, is genomic selection. So in genomic selection, we aim at using the genotypic data in order to predict the phenotypic performance of a population just using genotypic data. But for doing so, we have to have very good and reliable and robust genomic selection models. To build models, we have to have what we call training population. A training population has a phenotypic information, the data that we took in the greenhouse, in the field, or in the lab. And this population has also genotyped using different platforms. And we get, for example, molecular markers such as SNP. And then we use those data to train models, and then we get from the models what we call the marker effects, and we use those marker effects to predict the performance of a genotype that has not been phenotyped yet. And the advantage of genomic selection in a breeding program is that it will save a lot of time to the plant breeder, and it will increase the genetic gain per unit of time, but it depends on the trait and trait heritability and the models that the breeder are investigating. That's the brief technology that I think is useful for us before entering to the different sections of the presentation. I will talk about conventional breeding in Calpe. So this section has our examples of what I have been involved at the University of Arkansas regarding the Calpe breeding program. And for the program, my major professor and the program are interested in yield, like every other breeders. Yield is an important trait. And when I had my PhD candidacy exam, and one of my professors was asking me, if you're managing a breeding program one day, what will be list 3 traits that you think might be important in the program? And there was citing yield, disease resistant, abiotic stress, and those are good traits. But he said the most 3 important traits in a breeding program are yield, number 1, number 2, yield, and number 3, yield. And I think to some extent that professor is right. And at the University of Arkansas, we were also interested in Calpe that has erect type, because those are easy to harvest, and farmers can use combine for harvesting those Calpe. We were also interested in short season Calpe, and I remember we had some varieties that are mature for about 60 days, and those are very useful if farmers are interested in short season Calpes. We were also looking at some canning property of the peas, because some peas, when they soak into water and they put into the cans, some Calpe cracks. But our program was looking at some Calpes that have better property for canning purposes. And lately, there has been interest in developing Calpe for vegetables. Some people want vegetable Calpes, and they were eating the young green pods. And lately, there has been some interest in having high protein Calpe. And the program was in the process of developing high protein Calpe. And for the phenotyping, they have been using NIR and elementary rapid and instrument. So NIR is a machine where you collect the Calpe seeds, and then you put into the machine, and you have a percentage, the estimate of the percentage of Calpe protein within that seeds. But the elementary rapid and free instrument is we have to grind the Calpe and do all the lab work, and then use some formula to estimate the Calpe protein content. And the second method, the elementary one, is the widely used method, but the NIR is faster, but we need to do some calibration, because we have seen that if we estimate the protein content from red peas, Calpe, that are red type, the correlation was a little bit not very high if we correlate the results from the NIR to the elementary rapid N3 instrument. And we had to do calibration, and we had some paper on that work. So here I'll be presenting to you some of the cultivars that have been developed, and I was involved in the variety trials of those cultivars. And those cultivars are credited to the faculty at the University of Arkansas, and they have been working for the Arkansas breeding program was, I think it's over 50 years, and they have been developing about 20 varieties so far. And the time I was at the University of Arkansas, the program was releasing 4 varieties. And I will be presenting to you briefly the 4 varieties that have been released. And I would like to mention that there has, those cultivars have been developed without the use of any molecular markers, but it has taken a lot of time to get those cultivars out. And we have this first cultivar, which is a pink eye Calpe. And the second cultivar is a pink eye as well, but the eye is a little bit darker, and it is a little bit longer in terms of the maturity is a little bit longer. And the third one is a red type Calpe, and this is the one that I told to you before. If you use an elementary an IR machine to evaluate the protein content, you need some calibration, because the color of the pea will affect the protein evaluation. And the last one is cream type Calpe, and this is my favorite, because if you look at the pods, it is moving upward. It looks like a chicken feet. But if we look at those previous Calpe cultivars, the pods are moving downward, and it was a little bit special for me. And this is the conventional breeding for Calpe, some examples that I've been involved at the University of Arkansas. And I would like to mention that there has been any use of any molecular markers to develop those Calpe, but it took a lot of time. They have already started the process before I had joined the program. So now we have this technology called genotyping by sequencing and whole genome resequencing. We can use those technology to have the SNP markers. And the first one is genotyping by sequencing. This technology is relatively old, but it's still very useful. So we started using this technology on Calpe around 2014, 2015. And at that time, the Calpe genome was not yet available. So thanks to this technology, we can still get molecular markers without what we called a reference genome. But the downside is we will have a lot of missing data, and it can impact the downstream analysis when you do some genome-wide association study or any kind of bioinformatic analysis. So we started investigating salt tolerance in Calpe, and we have been using a relatively small population size. And we phenotype those Calpes in greenhouse for salt tolerance. And I am jumping into the process. we have found about 10 SNP markers that was associated with leaf score injury in Calpe. And that was the first molecular markers for salt tolerance in Calpe using genotyping by sequencing. And this work has been published in Theoretical and Applied Genetics. So there are also work that has been done for investigating low phosphorus deficiency in cowpea, and the phenotype has been done by our colleagues from Texas A&M years ago, and they have been using more than 350 genotypes, and we have the genotypic information on those cowpea genotypes, and this is the good thing that I would like to share with everyone. So if there is an available data somewhere, and if you have data that can complement this data, it is a good thing because you can work a little bit faster and generate result faster and get publication and abstract faster. And I think it is a good approach for a breeding program. And coming back to this low phosphorus deficiency tolerance, we found about 10 SNP markers as well that have been associated to this trait. And the detail about the statistical models and all the steps that we have been conducting to find those SNP markers are available on this paper. Plant growth habit, as mentioned earlier, is also an important trait in plant breeding. And the phenotypes have been done by our colleagues from Georgia, and we did the phenotype at Fayetteville as well. And we have also found SNP markers from GBS data that were associated with plant growth habit. So we have also done some work on iron deficiency in cowpea and find some SNP markers associated with this trait. So as of right now, we have found a lot of SNP markers associated with the traits. And what are the use of these SNP markers? So we can use those SNP markers for selection. So if you are targeting one trait, but you want also to stack those, let's say, genes controlling those traits into one cultivar, it is very useful to be provided with those SNP information data in a breeding program. And that's the step where we called marker-assisted selection in a breeding program, where we use those SNP markers and implement those SNP markers in the breeding program so we can make the selection a little bit faster and we can handle a larger population, because we know that field phenotyping is a very labor intensive, and we can decrease that labor using molecular marker. And if we have more time, we can increase the population, which can compensate the labor from field phenotyping. And that's the good thing with having a lot of molecular markers, besides the fact of having publications which are required. Genotyping by sequencing is great, but as I mentioned, we have a lot of missing data, which can make the analysis a little bit harder. Then we have used a new approach to find SNP, and that new approach is widely used, and it is called whole genome resequencing. For this project, we have a phenotype about more than 300 copy genotypes from different countries, and all materials that we have been using for further phenotyping and analysis were derived from one plant. Because when some of those seeds are from USDA, and within that the small envelope that you get from USDA, some seeds are mixed up, and when we plant within the field, we have seen a lot of segregation within the row. So we take one plant and then harvest the seeds from that plant and take the leaves for DNA extraction from that single plant. So the result will be much more accurate and robust. And the plants will be more uniform as well. So before I have been talking about the salt tolerance, but I was skipping the phenotyping at that stage. And on this slide, we have a more detailed about how the salt phenotyping was done. The challenge about phenotyping for salt tolerance is if you do the experiments in the field, we have a lot of uncontrolled variability, like the temperature and all uncontrolled variability that can occur in the field. So doing the phenotyping within the greenhouse is much more uniform. But again, the challenge is how we can translate the salt phenotyping from a greenhouse into field conditions. And that's another question. But for this research, we have been focusing on the salt tolerance within greenhouse conditions only. So for each of the materials that I presented earlier, we used 4 plants within small pots, and then we start treating the plants when the first trifoliate leaf begin to expand, and we've used this concentration of sodium chloride. We have done some preliminary work to identify this concentration, and we treated the plants from trifoliate leaf expansion until the susceptible control was completely dead, and the treatment was done for 2 hours every day. And for the experiments, we had a set that has been salt stress and another set that has been used as control as non-salt stress, and we just watered that set of plants using deionized water. And the process, the methodology for phenotyping salt stress in greenhouse condition for Calpe has been developed by our team, and it has been published as well. Now I would to provide you with some information about the phenotypic data that we have been collecting. And here I would like to mention that the phenotyping is the key for a breeding program to be successful. Everything is based on phenotype, and the tools like GWAS, genomic selection, molecular markers, these are tools that can help us develop cultivar faster. And I think that using those tools and the phenotypic data, we can develop cultivars faster. And those are 2 things that are complementary. So regarding the association, the phenotype salt tolerance phenotyping for the association panel, we have taken a lot of traits, but for today's presentation, I will just elaborate on leaf injury score due to salt stress, and also the chlorophyll that we can obtain using a SPAD meter. So we can see that the distribution of those traits are almost normally distributed. So it can suggest that Calpe can be controlled by many genes. Since because if it was a bimodal distribution, then there is high chance that this trait is controlled by 1 or fewer than 5 genes according to the quantitative genetic principles. And on the right-hand side of the slide, we can see how the phenotyping was done in the greenhouse. And using this methodology, we can phenotype a lot of breeding lines in a very short period of time. And that is really good because you can expand your population and do phenotyping in a little bit in a faster way, and we want faster results and also accurate result. And I think this approach is a good one to achieve this purpose. So we are still on the salt tolerance phenotyping side of the presentation. And up here we have different box plots. And for each box plot corresponds to one trait. And for due to time constraint, I will just elaborate about the leaf score injury due to salt stress. On this box plot, on the x-axis, we have the geographical areas where origins where the Calpe genotype came from. And we have AFR, which stands for Africa, AM for America, AS for Asia, EME for Europe and the Middle East. So we have grouped those genotypes into those geographical areas due to sample size limitation. If we split further the genotype, for example, from EME, the Europe and Middle East, we won't have enough sample size within that group to do a more accurate statistic. So getting back to this box plot, we can see that the genotypes from AM, America, have a lower leaf score injury. And for the leaf score injury, it went from 1 to 7. For 1, we don't have any chlorosis or necrosis appearing on the leaves. And for 7, the plants were completely dead. And we can see that the genotypes from America have a lower leaf score injury, meaning that they were likely to be more resistant to salt stress compared to other cultivars. So we can suggest that those lines could be used as parents for developing a salt tolerance population for breeding. And I would like to mention that most of those lines from America were breeding lines from University of Arkansas. And currently, myself and the resident director are working on some MTAs in order to test those lines into the Texas A&M conditions. And we'll see if some companies also are interested in the lines as well. So now I am moving to the whole genome resequencing approach that we have been doing. So for this study, we have obtained about more than 14 million SNPs from the company, and we did some filtering because not all SNPs are suitable for analysis. And after SNP filtering, we have about slightly less than 6 million SNPs for conducting the Genome-Wide Association Study, or GWAS. And we can see the distribution of those SNPs on this chart, with chromosome 4 and chromosome 10 having the highest number of SNPs, and with chromosome 2 and chromosome 7 having the lowest number of SNPs on average. And it is also, the number of SNP per chromosome is somewhat correlated with the chromosome length as well. Sorry for that. So we have used those SNP information to conduct a genetic diversity and population structure. And so far, I couldn't find a way to use all 6 million SNPs to run this analysis. So I selected 60,000 SNPs to conduct first the population structure and then genetic diversity analysis. And the population structure analysis using those 60,000 SNPs take weeks to run. And after the analysis, we take what we called cluster or groups or subpopulation information, and then we try to combine it with genetic diversity analysis that was run in a different software. And the combined analysis is shown using the figure that we can see from this slide. And the interesting thing is that the breeding lines from Arkansas were grouped together. It means that those lines were, the pedigree of those lines come from common pedigree, and it makes us also comfortable because the data should be accurate, since those lines that come from one breeding program were clustered together. Now I would like to provide you also with some information regarding the Genome-Wide Association study on salt tolerance on cowpea. And this one was the leaf score injury that we have been talking before. And we have what we call a Manhattan plot. On the x-axis, we have the chromosome information. And on the y-axis, we have what we called LOD. The higher that value is, the better your SNP is. And it is there is a higher likelihood that the SNP is associated with your trait of interest. And the most interesting thing that we have found is that we found that there is one SNP that was very close to a potassium channel. And we know that potassium channel can regulate salt stress in plants, like in Arabidopsis, for example. And we were very pleased with those results. And the next step is to validate these genes. But the issue is the transformation protocol for Calpe is there are protocol transformation for Calpe, but it is still challenging. So if you want to study the function of these annotated genes, you have to do it in another crop, like in Medicago, or I am not sure about soybean, but maybe Arabidopsis as well, if it won't work for Medicago, which is also a legume, and it is close to cowpea. Now I would like to provide more information about how we can use those results in a breeding program. So we have one SNP that we found from the GWAS, and then we want to have more information about the alleles that are beneficial to that trait. For example, we want some alleles that can reduce the leaf score injury in cowpea. And for this result, we found that for the SNP under this box plot, the allele TT could be beneficial in reducing leaf score injury in cowpea. So in a breeding program, we can use this allele for selection. Let's say we have 1,000 plants, and we select the plants with an allele C at this position. And that's how we can use those results in a breeding program and make the, make the selection a little bit faster, and it will save time. We have done also some work on drought tolerance phenotyping, and this methodology was developed by Dr. B.B. Singh, and we are very grateful to him, since this approach has saved us a lot of time. So prior to conducting these experiments, I did some experiments in the field, but it was very raining at that time, and there is no way we can do drought tolerance phenotyping in the field condition. So thanks to this methodology, we can do some selections at seedling stage in greenhouse conditions. But again, the question is how we can translate this information into real field conditions, like at Vernon, which is an excellent site for doing this kind of analysis on the field. And this is one of the project that I'm looking forward to continuing, about doing field screening of drought tolerance in Calpe at the Vernon Center for next spring. And I am going back to the result from this study. We have taken a lot of traits, the more the better. But for today's presentation, I will just focus on drought score and recovery after watering. For the drought score, it was a scale going from 1, the plants were fully green, and 5, the plants were completely dead under drought stress. And we can see from the figure on the right-hand side of the slide how the experiment was conducted. And at the bottom, D, we have a very clear discrepancy between a genotype that is fully green and a genotype that is fast wilting under drought conditions. And these are excellent phenotypes if we do screening, because you can do it very fast and you can screen a lot of plants in a very short period of time. And if we do scoring of those plants, we can have this histogram on figure A. And for the population that we have been investigating, the distribution was approximately normal. But for the plant recovery, it was skewed. Not many plants were able to survive. So after the susceptible genotype was completely dead, we then rewater and look at which genotype among the dead ones were able to be recovered, and not many according to our observation. Here we have a similar analysis to what we have been doing for tolerance in Calpe. And again, we have this box plot showing the variation among different areas in terms of drought score in Calpe. And we can see that there was not too much variability among the lines from America, Asia, Europe, and the Middle East. But surprisingly, the genotypes from Africa, AFR, were a little bit drought susceptible, and I was a little bit confused with this result, but I think that the lines that we have been using for Africa has already bred for other things, maybe for pest or disease resistant, but we didn't have information on that aspects. So I just leave this result as it is, and maybe you can also have some suggestions on why the lines from Africa were a little bit drought susceptible compared to the lines from elsewhere. We also did some genome-wide association study analysis, and the most significant finding was this huge peak at the start of chromosome 5, and that was a trait on trifoliate leaf tolerance. And this trait was also investigated in some Calpe lines at the Texas A&M University, and a grad student, I think, named Dr. Verby, has using this trait, trifoliate leaf chlorosis, to investigate his population. So we were inspired by his phenotyping and did the same phenotyping for our population, and we found this huge peak at the start of chromosome 5 and some candidate genes that were worth looking as well. We have also some result about multi-parent advanced generation intercross, or MAGIC, and those lines were from UC Riverside, and we are very grateful to those scientists who have been providing the lines to us for free, and this is how the lines have been developed. I am jumping directly to the result about the salt tolerance for this MAGIC population. Remember, I talked about an association panel before, which is one population, and now I am talking about a MAGIC population, which is a different population. And the good thing with a MAGIC population is that it makes the allele more balanced. And if you do a genome-wide association study, GWAS, there is a good chance that you can find the rare alleles associated with the traits. And we know that some of of the useful traits in agriculture can be associated with rare alleles, and we can easily miss those alleles if we use a random population. And that's why we have been also using the magic population. And on the histograms that you can see from the figures on the slide, the parents from which the population was developed are diverse in terms of leaf score injury and spot chlorophyll, for example. And this variation is indicated by those black arrows. And on the right-hand side of the slide, you can see how the phenotyping was conducted. We took a lot of traits as we did for the association panel, and we displayed the correlation between traits using a circus plot, and some traits like chlorophyll content under salt stress was correlated with number of dead plants, which makes sense because if the leaves are yellow, they were almost dead. But the plant height that we took under salt stress was not really correlated with other traits that we have been taking for this study, since the wider the link between traits are, the more correlated the traits. And you can see that the links for plant height was a little bit tiny, so there is weak correlation between reduction in plant height under salt stress with other parameters such as number of dead plant or leaf spot chlorophyll. And that's the bottom, the most important finding that that we have found regarding salt tolerance phenotyping in this magic population. And on the right-hand side of the slide, this shows the different overlapping SNPs between traits. And as we can expect from the correlation study from the circus plot, we don't have correlation between number of dead plants and the number of the reduction in plant height due to salt stress. So we don't expect having significant SNPs for those two traits, unlike other traits. And up here, this is just a summary of the SNPs that we found. The LOD is a little bit low because this magic population was first genotyped using a 50K SNP as compared to the 14 million SNPs that we have before. So this is already expected. But the good thing is, despite this moderate LOD value, it means that as I mentioned before, the higher your LOD is, the better your SNP is. So despite this moderate LOD, we can still find some genes that have been involved in ion dynamic in plants. And here is the epistatic interaction modeling. It is a way to understand how the different SNPs interact between themselves in order to be associated with some traits. And we found some interaction between different chromosomes, but we hardly find interactions within chromosomes. And I don't have an explanation about why that is. And we did some work about genomic selection as well for both association panel and magic population. And this one is the result from the magic population, and the genomic selection was about, accuracy was about 0.5, and the different colors just correspond to different training population size. We have also looked at the maturity date under drought stress and how that maturity changed using this magic population. And we found significant peaks as well. regarding grain yield reduction due to drought stress using this magic population, the LOD was a little bit low, and it was also expected since grain yield was is a polygenic trait. As I mentioned before, we have a lot of traits, and we were interested in understanding how the traits were correlated at the genetic level. And this is a result that we found. We found that there is some correlation between grain yield and plant growth habit under drought stress at the genetic level. And this work is almost published in horticulture research. So to sum up, conventional breeding is great, and we have to do conventional breeding, crossing, phenotyping, but it can be accelerated using molecular breeding. So those two concepts are complementary. And we have seen also that using a whole genome resequencing approach, we can find candidate genes and then do some functional genomic works later on those. The information from those candidate genes can also help breeding as well. And we have been talking about the process of developing molecular markers in Calpe. And for Calpe, not many molecular markers are available, unlike crops like soybean and corn. And we are in the process of making those molecular markers a little bit available to everyone. And for the future work, since we have some good genotypes from the University of Arkansas, so I will be planting those genotypes under organic systems in some farm, in some sites at Texas, since my position is also about organic breeding. And we are also in the process about working on MTA, about using the lines from Dr. B.B. Singh to test, to conduct the evaluation under organic farming systems for next spring. And the purpose I am currently developing is that we can have both vegetable, seed, and forage type cowpeas, but I will focus on the seed cowpea because those are one of the type of cowpeas that the industry are interested in. And obviously work related to drought, heat, and salinity need to be conducted as well. And we are currently responding to a proposal for FFAR for heat tolerance in Calpe. And on that proposal, we will also include the feasibility of UAV phenotyping. And I will, regardless of the outcome of the proposal, we will be doing some work on UAV phenotyping for various traits in Calpe because this will help a lot on the phenotyping aspect. And in order to make this program competitive, I will include epigenetics. So because this is something new, and if we can incorporate something new to the program, it will make the program a little bit competitive in terms of grant proposals. Because if we just focus on cultivar developments, it's very hard to find some innovation on that area, and we can incorporate more things. So these are some selected papers I have used during the presentation and some abstract. And I would like to thank all the attendees for your time and consideration and your support. And I am very fortunate to have this opportunity to provide my background and some perspective regarding cowpea breeding and genetics to you. And I'll be happy to answer any questions if you have. Thank you.
Excellent job. Very impressive work. Questions?
Thank you, sir.
Hi, I have a question.
Yes, sir.
Hi, great presentation, Dr. Waltram. And
thank you.
I'm curious to ask you, So you used two different populations and you ran some QTL analysis and you found some candidate genes. My question is, those candidate genes are related in both populations? I mean, those candidate genes are somewhere close together if you compare the magic population and the other population that you used?
Yes, that's an excellent question. So we have take a lot of traits, and there is one trait. This is chlorophyll, the spike chlorophyll under salt stress, and this is for the magic population. And if you look at chromosome 2, and I will go back to the salt, the GWAS for salt tolerance, and on chromosome 2, at the end of chromosome 2, you still have this peak, and those peaks were within a 700 kb region. And that's the kind of overlap we find for salt tolerance in Calpe. But for drought tolerance, the drought tolerance one was conducted at the UC Riverside under field condition. But the phenotyping that we did for the association panel was in a greenhouse condition at the University of Arkansas. But unfortunately, we didn't find any overlap between those two population in terms of drought stress, but we find some overlap for, I believe, 1 or 2 traits for salt tolerance, and this is an example of the overlap that we found. Does this answer your question, sir?
Yes, yes, it answers it. Thank you very much. Again, thank you for your presentation.
Thank you.
Oh, there's a question in the chat. I'll ask it for Shakira.
Thank
Great presentation. Are there any yield advantages or better stress tolerance of the pink-eyed Calpe over the black-eyed Calpe or vice versa?
Oh, that's a really good question. I should have run the analysis comparing the different Calpes in terms of color or type, like black eye versus pink eye, but I haven't done this yet, but I will do it. So I don't have information yet on that. And I think that's an excellent question as well, like to see the correlation between the color and the type of Calpe and their yield advantage and performance under stress. Thanks for the note.
Sure. Other questions?
So what do you perceive to be the big market for Cowpea?
Thank you for the question, Dr. Baltensberg. I think, so recently I've discussed with a company called the New Green Deal, and they are interested in seed Calpe with a better, with a bigger seed, and also short season Calpe. And I think it applies to most of Calpe industry about having some genotypes that are short season, having bigger seed, and also having some adaptability to where those Calpe are grown. And I would like also to add about organic Calpe. This can also be a market for the Calpe breeding later on, since to the best of my knowledge, I am not sure whether there are organic Calpe provider, organic seed Calpe providers in the company, and I think that could be a great opportunity as well. Does this answer your question?
Yes.
More questions? Go ahead, Amir.
Yeah, thank you so much for the great seminar. You talked about the salt tolerance SNPs. If you want to convert these into Cas-based markers, we'll be glad to help you in the agri-genomics lab that I co-direct with Dr. Stelly. But you know, also Dr. Shuyu, when you came for the interview, also provided this invitation that you can use his lab in Amarillo.
Yes.
So our lab can help you with that. And the AGL lab actually works on membership. So I will be contacting you to express that and to show you how we can help.
That would be great, and thanks in advance for that.
Sure.
And do they have a little bit of discount?
Well, actually, we will talk about the details later about that. So I'll call you. Okay.
Okay.
Thank you so much. Thank you.
Thank you, Dr. Ibrahim.
You bet. My pleasure.
Other questions? You know, legumes have carbohydrates that are not easily digestible by humans, but the bacteria in our guts seem to really feast on these things. Is there any like proposal to search for legumes with less of these insulting carbohydrates, for instance?
I am not very familiar with that area on carbohydrates. And this is a new information to me.
Okay.
Thanks for that note, and I will keep that in mind.
Okay.
So
other questions?
For Calpe, we usually focus on proteins.
Yes, I understand.
And carbohydrate is in fact, there is a study about sugar, and some people want some sweet Calpe, and this could be an opportunity, a niche market as well.
I see.
But I am not sure about the genetics behind carbohydrates in legumes and like Calpe, for example.
Dr. Zhang, did you have something? Go ahead. No, your microphone isn't on, Dr. Zhang. Dr. Zhang, your microphone is not on. We can't hear you. You could type the question in the chat.
Let me walk to his room to tell him that.
Okay, thank you.
Thank you, Dr. Ibrahim.
Okay. There we go. It's in the chat. So I'll ask it.
Okay.
Dr. Zhang says, you mapped the salt tolerance with two types of populations. Which one got more SNPs for the trait?
The association panel. Because we have genotyped this population using 14 million SNP.
Okay.
But for the magic population, we have had the SNP data, 50K SNP data, and the difference from 50,000 SNP to 40 million SNP is huge. And obviously, we have a lot of SNPs from the population that have been phenotyped with 14 million SNPs. And I have not developed about the statistical models, but we have already done a lot of work on reducing the false positive discovery on using those this large amount of SNPs.
Okay. Does that answer your question, Dr. Zhang?
Go ahead.
I said you map the kid tolerance with the salt tolerance with two types of population. One that's regular traditional GWAS panel, another one that's magic. My question very simple: which one you get more SNPs associated with the tolerance?
Of the association panel.
Association panel.
Yes, sir.
So another question: what's the advantage to use develop a magic population? You have a traditional GWAS population. Why develop a magic population?
Oh, that's a
resources to develop that population.
Yes, that's a good question. Like the idea was first, the salt tolerance study was not done yet for a magic population in Calpe, and also for a magic population, regardless of the crop, the allele is more balanced, like in a QTL, a biparental population. And if you have a more balanced allele, it's it will be a little bit easier to find the rare allele. In contrast to an association panel, where if you have an allele that occur 1 in 1,000 individuals, so it's a little bit harder to find that rare allele in an association panel.
Yeah, it's fair. I know quite a few crops use the magic population. For example, corn, the product get an NSF, that's $5 million to develop that population.
Oh,
yes. I understand actually there's a disadvantage use the magic population relative to traditional GWAS. What's the advantage GWAS over the Cowpea mapping? That's they lose that point advantage.
Right.
For single traits, use the traditional GWAS population, you should get more Cowpeas.
Yes.
Based upon the genes compared with magic, compared to Cowpea mapping population. So that's why I say why people do that.
Yes.
The same thing for coffee. It's good job, but you good talk. That's my question here.
Yeah, that's
because
a very
both population and the population.
Yes, that's a very good question, Dr. Zhang, and I have just like a very quick comment. So like this magic population was developed by the UC Riverside and founded by USAID and got significant amount of grants.
The parents are 5-6 lines. You use 331 lines. That's genetic variation much broader compared with the magic population.
Right.
Your lines, their lines are originally use 5 lines. Your panel is 331.
Right.
Worldwide representative, right?
Right.
Okay. Thank you very much. Good job. Good work.
Thank you. I'm looking forward to working with you as well, since I think you have.
I work with a little bit with Dr. Sen in Calpe as well. We work on
yes, I read some of your
finger tolerance and the drug tolerance we're doing. We done the job a few years ago. Student graduate that is writing paper right now. We start. It's okay. You do yours. That's good. It's not. We work on the gene-based breeding. We also do genome selection.
Oh.
Our major focus is the gene-based breeding because much powerful than genomic selection.
Yes.
Yeah. We're writing a number of papers now. Okay. We're looking forward to work with you in Calpe.
Yes, Dr. Zhang.
Okay.
Are there any other questions for Dr. Ravelombola?
Thank you so much.
Well, I'd like to thank you again for a wonderful presentation.
Thank you, sir. And just a quick comment. I have you got A++ in pronouncing my last name because most of people were struggling pronouncing it.
It's not that difficult.
Thank you so much.
All right. And we'll see the rest of you back next week, same time, same channel.
Scott, do we still have a date we're short?
No.
Great. Thanks.
Yep.
Thank you all.
Bye-bye.